This method was designed to quantify contact-dependent competition via the type 6 secretion system between bacterial strains at the single-cell level. This technique uses total cell area rather than cell counts to quantify bacterial competitions, making it easy for researchers without proprietary image analysis software access. This method can be easily modified to quantify competition between diverse, culturable microbes. Additionally, the experimental setup can be optimized for use on either upright or inverted microscopes.
To begin with, prepare the agarose pad solution by dissolving 2% low-melt agarose into marine PBS. Heat the solution briefly for about 30 seconds in the microwave, and vortex until the agarose is completely dissolved. Keep this solution in a 55 degrees Celsius water bath until ready to use. Next, wrap a piece of lab tape around a glass slide 5 times. Repeat wrapping of tape on the same slide such that the distance between the 2 tape pieces is slightly smaller than the cover slip width. To ensure a flat agarose pad surface, pipette warm agarose solution between the 2 pieces of tape and immediately put a cover slip on top such that the cover slip makes contact with the liquid and pushes out any bubbles in the agarose solution. Let the agarose pad solidify at room temperature for at least 1 hour. Then, cut this agarose pad with a razor blade into 4 5 square millimeter pads to be used for imaging. To prepare the strains for co-incubation, streak out each strain from frozen stalks onto the LBS agar plates, supplemented with the appropriate